Pathology, apoptosis. All right, so when discussing cellular death, the number one thing that we need to discuss is the difference between apoptosis and necrosis. This will specifically focus in on apoptosis, and the next video we'll be talking about necrosis. So delving into apoptosis, what we first need to understand is that this is an ATP-dependent programmed cell death. So this is essentially cell suicide, okay? And a very key concept about apoptosis is that it's singular cell death. It's not massive amounts of cells dying altogether. It's just one single cell programming itself to die. It's not followed by any inflammation, unlike necrosis, as we'll see, okay? And apoptosis can be pathological, but it can also be a part of just normal physiology, okay? And that's an important aspect that we need to understand. Lastly, a weird concept that they like to test on is that for DNA laddering, fragments in multiples of 180 base pairs is an indicator of apoptosis. So if you ever see the number 180 for 180 base pairs when they're talking about DNA, start thinking about apoptosis, okay? Although relatively low yield, that could be a question that comes up. Now, there's three different pathways in which apoptosis can occur. The first pathway is known as the intrinsic pathway, also known as the mitochondrial pathway. And the reason that this is known as the intrinsic pathway is because the mitochondria is located within the cell. So the response to which we should go under apoptosis is coming from the mitochondria, which is coming from an intrinsic signal because it's an intrinsic organelle, okay? So that's why they call it the intrinsic pathway. So I gave you already a little hint. This is the mitochondrial pathway. That means the sign in which apoptosis is getting sent to, right? So the signal to the cell that we should program death for apoptosis is coming from the mitochondria. So let's begin. There's multiple key players for the intrinsic pathway, but there's three that I really want to focus in on. The first one is called BAX and BAK, okay? So BAX and BAK, BAX and BAK, these things are pro-apoptotic signals, okay? So these like death, okay? So BAX and BAK like death. And what they're constantly trying to do is that they're constantly trying to get cytochrome C to be released from the mitochondria and go and travel to activate caspases, which in turn leads to apoptotic death, okay? So do we understand that process? So that's the first aspect of this. BAX and BAK, BAX and BAK, are constantly trying to go and allow cytochrome C to be released from the mitochondria so that cytochrome C can go activate caspases and increase caspases, and caspases in turn go and activate apoptosis, all right? Now, the question is, why don't we all have apoptosis constantly in our cells and it just leads to the destruction of our whole organism? Well, we have the second player and a very important player. It's known as BCL2 and BCL-XL. So BCLs are very important because what they do is that they inhibit that BAX and BAK activating on cytochrome C, okay? So they inhibit the release of cytochrome C from the mitochondria, making them anti-apoptotic, okay? So very important to understand that. BAX and BAK want cytochrome C to be released to activate caspases, which leads to ultimately apoptosis. And BCL2 and BCL-XL, the BCLs, block cytochrome C release, okay? Very important, all right? So there's something that ties into pathology that we will discuss in the hematology oncology section later on that plays a role in this BCL2 as well as a BAX pathway. So BCL2, as we said, is anti-apoptotic, okay? And at times, the BCL2 gene, okay, it can be overexpressed if there's a translocation between 14 and 18, chromosome 14 and 18. This can lead to follicular lymphoma, okay? And the reason behind that is because now we will constitutively express BCL2, which is once again anti-apoptotic, leading to a decrease in caspase activity, causing decrease in apoptosis and an overall increase in tumor genesis, okay? So potential for cancer, specifically follicular lymphoma for T14-18, all right? And we would discuss that further in detail for the hematology oncology section, okay? Next up is the extrinsic pathway. This is known as the death receptor pathway. And the reason it's extrinsic, once again, is because there's some cell coming from the outside and signaling for our own cell to undergo apoptosis. Okay, so what happens is that we have a cell that has a ligand receptor function known as FAS-L. Okay, so FAS-L binds to FAS, F-A-S, CD95. Okay, in order to activate programmed cell death and leading to caspase release and apoptosis ultimately. Okay, normally this FAS-L interaction is seen in the thymic medulla for negative selection, and we'll talk about it for usually in our immune section. But for our immune section, we're just going to just really quickly, negative selection. All that it is that when we create our own immune cells, we want to make sure that it can positively select and negative select, so it can bind to an antigen, but it can't bind too hard to our own self antigens. And so if it does, then we do negative selection, which ultimately causes the death of the cell that binds too hard, and that's known as negative selection. Okay. Now, what happens if there's a defect in this FAS-FAS-L interaction? Well, then we can get ALPS, autoimmune lymphoproliferative syndrome. All this is that we have a failure of that FAS-FAS-L interaction, which leads to an inability to have negative selection. So we have an increase in number of cells that are self-reactive to our own antigens. This can present with lymphadenopathy, hepatosplenomegaly, and usually autoimmune cytopenias. Okay. So the way that I remember this is that you go FAS on the ALPS. So FAS-L pathway is going to be in charge of, or usually the pathology associated with, autoimmune lymphoproliferative syndrome, also known as ALPS. Okay. Lastly, and the last pathway for apoptosis is the perforin granzyme pathway. Okay. And all the perforin granzyme pathway is, they're kind of well named because what it does is that you'll have the release of granules containing perforin and granzymes from autoimmune cells. Okay. So from immune cells. And what they do, the perforins are going to perforate, okay, so make a hole in the cell membrane of our cells. And then it throws granzymes or grenades into the cell of choice that it needs to undergo apoptosis, leading to the trigger of cell death or apoptosis, okay? So that's how I remember it. I remember it as perforate and then throw in a grenade, so perforin, and then granzyme for that pathway, all right? So overall, what you need to know about this is the intrinsic mitochondrial pathway involves BCL2 as well as BAX, and BAC for cytochrome C release. That's what they're fighting over, which leads to caspase activation and apoptosis ultimately. And the pathology associated with an issue with that is going to be follicular lymphoma, which is translocation of 1418 because of the BCL2 overexpression. Next is the extrinsic pathway. What you need to know about this is that this is the FAS-L pathway, and any defect can be associated with the pathology of ALPS. And lastly, the perforin granzyme pathway, all you have to know is that it uses perforin to open a pore into the cell membrane, and then it'll throw granzymes in there in order to destroy them, okay? That's all for apoptosis.